Good morning, everyone. Thanks for coming out this morning for this lecture. Well, lecture slash theory of practicals. Challenge, or the reason why we're having this particular 513 class as alternative to practical is because the equipment required, a dissolution tester, is not currently operational. We're hoping that it will be repaired as soon as possible, but then because of logistic reasons, it's not yet been made operational. So as soon as, updates will be provided to the class in time, as time goes on. So the first part of this class deals with generic substitution, and then the second part deals with in vitro testing to prove bioequivalence. Both of them are based on very similar principles. For example, generic substitution will not happen if we don't establish pharmaceutical equivalence and bioequivalence. And similarly, the in vitro testing happens because we need to prove the bioequivalence of a medicine. So the reason why we'll go this route of first doing generic substitution before we look at the in vitro testing is because not all medicines require, not all medicines will pass for under the BCS classification for in vitro bioequivalence proof of their bioequivalence. Some of the medicines that we use, especially biologicals and large pharmaceutical molecules, will require actual in vivo, that's in human trials, clinical trials, to show bioequivalence. But one of the bastions of generic medicine substitution in clinical practice is because generic medicines are developed as copycats. So they copy what the innovator medicine has done. So they have a shorter time on rail to get that from research to bench trials to to human trials. It's a very shorter ramp than for the innovator medicine. Innovators medicines take sometimes an average of 10 to 12 years before they come to market, except with the innovations around what's it called now? Innovations around testing and the use of AI to simulate what medicines will act like, in silico trials, right? So aside from that, the usual time for a new medicine to come from identification in the lab to human trials takes about 10 to 12 years. Now, because of that, innovator medicines are very expensive, and usually to allow them to recoup their money, they're given a patent period of about 10 to 12 years as well, which allows them to market exclusively, sell exclusively, and then at the end of that period, right, the patency expires, which means other people would have also obtained those medicines when they were under patent and began to reverse engineer those drugs, right, until they can also come to market once the patency of that medicine expires, right? So that's the way, that's the main reason why innovator medicines are expensive. A lot of research, a lot of expenditure going into educating professionals, healthcare providers, including lobbyists as well, and registration costs. All of that takes a lot of money, right? So, for example, a clinical trial will cost between 50 to between 50K, 50,000 US dollars to as much as sometimes 2, 3, 4 million US dollars, right? So the idea is to allow pharmaceutical companies recoup their investments before other people are allowed into the fray. Now, even when other people are allowed into the fray, innovator products still remain very expensive above their generic alternatives. And during this class, we'll see why that is so. Short outline. Now, in terms of, by way of introduction, medicine product selection is a core responsibility of pharmacists, right? And that must be done with all sense of responsibility, because the outcome of treatment will depend on the type of medicine selected. Now, medicine selection is in basically two phases. It's on the side of the professional in relation to the supply chain. Medicine selection means that the pharmacist, has limited resources, so they choose medicines to stock, right? They cannot stock all alternatives. For example, for omeprazole, right, there is the innovator product and Mepracil, and then there are the generic products of different names, right? A single pharmacy cannot afford to have all generics of omeprazole, so they must use their professional distinction to select alternatives to Mepracil to hold on their shelf. Now, medicine product selection in service to the patient is, for example, choosing between the innovator product or a generic, right, to provide to the patient. So let's quickly look at some of the evidence of why we do what we do. So I mentioned a number of terms at the beginning: pharmaceutical equivalence, bioequivalence, and pharmaceutical alternatives and therapeutic alternatives, right? Now, for drug products to be considered as pharmaceutical equivalents, right, it means those drug products are considered to have the same active pharmaceutical ingredient, API. They must be of the same dosage form and route of administration as the innovator product. So, for example, they are both tablets, and they are both taken per oral, that's through the mouth, or they are both injectables and injected in the same way, for example, subcutaneously, right? They meet the same compendia or other applicable standards for purity, identity, strength, and quality, right? Of course, as pharmacists, all of those applicable standards are in the compendia, right? Even though they may differ in shape, the way they are scored, excipients, and other things, if they are similar in all of these four forms, then we accept them as pharmaceutical equivalents, right? Alternatives, of course, there's no need to prove. What we just need to say is that they contain the same therapeutic moiety. That's the same active pharmaceutical ingredient, right? That's what we mean by therapeutic moiety. There may be different salts, esters, or complexes. For example, there's tetracycline hydrochloride, and there's also tetracycline phosphate, right? There's also erythromycin decahydrate. There's erythromycin, what's the other one that's lighter in color? I'll try and recall the name, right? So there are different salts of the same medicine, okay? They are pharmaceutical alternatives. In fact, in most cases, you'll find that a lot of physicians do not even know the names of the salts. Well, when you get into practice, you will find that a lot don't even know the strengths and how the dosage of the medicines should be applied, right? So it's important to keep that in mind so that when you begin to practice, as you practice generic substitution, you keep it in mind that you have to educate the patient when you're performing that professional duty. So the second important classification, the first important one is pharmaceutical equivalence. Alternatives, like I said, they're just in terms of salt changes, right? Sometimes these salt differences may affect solubility of the API, but in cases where in some cases you have the salts being able to help. So some manufacturers develop a different type of salt for a therapeutic moiety, so that their what's it called, their fillers, right, that are added to the APIs during compounding, right, do not affect the solubility or the plasma level of the active pharmaceutical ingredient. So sometimes those pharmaceutical alternatives are created in the service of ensuring that they avoid legal consequences, so that nobody says you copied my formulation, right? So even if patency has expired, you must do something that ensures that your pharmaceutically equivalent alternative, right, is functionally different. You know, you do not copy the design, the shape, the color exactly of the innovator product. So bioequivalence means that a medicine is pharmaceutically equivalent, right? And it has been proven to have the same biological activity, right, under the same experimental condition. So we'll say drugs are pharmaceutically equivalent when, in terms of their peak Pmax, Cmax, and Tmax, and total exposure, that's the area under the curve, after administration of the same molar dose under the same conditions, are similar to such a degree that their effects can be expected to be essentially the same. So it means that a generic drug product must be bioequivalent to the comparator innovator brand. Okay, so it means that when you use a cheaper alternative, it has the same biological activity as the generic medicine. So this negates all the arguments that cheaper does not mean better. At some level, that can be true, especially in our local context where we have significant substandard and falsified medicines in our supply chain. But on the other hand, if you're holding a generic product that is from the approved drug distribution system, then you can confidently say and assert that generic product is bioequivalent and so equal to the innovator product. Now, in terms of therapeutic equivalence, this is important, but you see, substitution on the level of therapeutic equivalence is usually not done alone. Usually when you do therapeutic substitutions based on therapeutic equivalence, you are required by law to inform the the physician who has recommended the medicine to the patient, right? So for therapeutic equivalence, medicines have to be drug products are considered to be therapeutic equivalent only if they are pharmaceutical equivalents for which bioequivalence has been demonstrated. So they have similar. Oh, sorry. That explanation was for therapeutic alternatives, not equivalence. So therapeutic equivalence really is just another word for bioequivalence. Sorry about that. That was ultimately wrong. So that's for when you're doing therapeutic alternatives, when you're switching medicines for therapeutic alternatives. For example, patients who have been placed on centrally acting antihypertensives like methyl dopa, right, are sometimes switched to better antihypertensives like ARBs, angiotensin receptor blockers, or ACE inhibitors, right, on the advice of their pharmacist, but of course with congruence, that with agreement of their healthcare provider, their physician, because of the added benefits that giving ACE inhibitors and ARBs provide to the patient. So for example, global benefits for ACE inhibitors and ARBs include renal protection, hepatic protection, and major organs and long-term survival over the use of just vasopressor drugs like centrally acting agents like methyl dopa that do not confer any other benefit apart from blood pressure reduction, right? So that is therapeutic alternative, that you switch to the same therapeutic class, but different therapeutic moiety, right? So here is therapeutic equivalence, which is the same for all purposes and intents as bioequivalence. Now, let's quickly talk about generic medicines, and they have a few definitions. The EMA, that's European Medicine Administration, defines it as medicinal products which has the same qualitative and quantitative composition in active pharmaceutical ingredients and the same form. That's the same. It's either tablets similar to that of the innovator, or syrup similar to that of innovator, or injectable similar to that of innovator, whose bioequivalence with reference medicinal product has been demonstrated by appropriate bioavailability. Even though it says bioavailability, right, the studies are usually referred to as bioequivalence studies, okay? Now, this is a reference. Now, NAFDAC did not require those bioavailability studies in the past, but ever since we've adopted this requirement for bioavailability studies in the recent past, almost about 10 years now, we have become a similar string. Sorry, no, not up to 10 years, it's about about 7 years now, right? We have become a similarly strict regulatory agency environment, similar to the European Medicines Agency. And we're on WHO class 3. Just yesterday, it was announced that the Central Drug Laboratory of NAFDAC has achieved pre-qualification, WHO pre-qualification status as well. So the next level will now be to evaluate, I think in the next 2 years, to evaluate the medicine regulatory strength and approve Nigeria for a tier 4 WHO stringent classification, which is the highest tier possible for medicines regulation. Right. Now for FDA, evaluation is based on suitability, therapeutic equivalence, as bioequivalence of generic drugs on scientific evaluation, which is similar to what happens in Nigeria now. Now, generic drugs are the same thing as multi-source drug products, right? And of course, the definitions are the same. Now, even though sometimes some people say that they are not interchangeable, but based on the bioequivalence being established in all the regulatory environments, generics are directly interchangeable for their innovator counterparts. Now, there have been concerns really that have been raised by, especially in the early days when generic substitution, began to become a thing, especially because of rising costs of healthcare and all. Concerns were raised about the kind of excipients. For example, there was, let me see if I can play a video for you. just to highlight some of the challenges that we're seeing, or maybe I'll send this later. Is your audio cracking? Can you hear me clearly? Is this better now? Shewa, you did not respond. Okay, the audio is not cracking. Okay, maybe it could be due to your own speakers. So, the concerns that were raised include that different excipients may be in different products. The excipients may be different from that in the innovator, and then the manufacturing process may not be as stringent as that of the innovator product. But of course, as long as manufacturers stick to current good manufacturing practice, all the other as far as the regulator is concerned, you have met what is required to produce a medicine that is, bioequivalent. That should be, pharmaceutical equivalent, right? But bioequivalent will need to be proven, right? And that this concern says may affect safety and efficacy of finished products. So this was proven in, I think it was the Netherlands. There was some patients were switched from Coumadin, that's phenytoin, phenytoin innovator product, right, to generic phenytoin, and it was discovered that some people were having too much the clotting factor, right? Some people were having hemorrhages, which were signs that they were already on an overdose of phenytoin, right? And then, not hemorrhages. Some people were having higher doses of, it was warfarin, not phenytoin, warfarin. So some people were having hemorrhages, right? And their blood was not clotting as fast as it should during tests and laboratory confirmations. And what was discovered was that one of the manufacturers changed one of the excipients, right, that led to increased levels of warfarin in the blood, right? And in some of the patients who were not responding to the innovator product, placed on this new formulation, they were doing well, but every other person that was doing well on the innovator product began to have this side effects of, hemorrhages. So what they did was they identified what was happening, and then they definitely identified that both drugs were no longer bioequivalent, since that excipient was causing a significant change in the biological activity of the medicine. So after that, it was demanded that all generics should begin to undergo bioequivalence studies, especially when the excipients may be non-inert. And then, of course, there's a widely accepted method to demonstrate that the differences have no significant effect on the ultimate product performance, which is the bioequivalence trial. So there are two ways to carry out the bioequivalence study. One is in vivo, which is human trials, and the second is in vitro. And there are different conditions under which both are demanded, right? Once you can prove either through in vivo studies, right, or through in vitro studies that your products are bioequivalent, then your products become interchangeable. If you do not prove that, right, and there is no use case for your medicine product, then that medicine product will not be considered as interchangeable or registrable until you can prove that there is a specific need, right? For example, in that warfarin product, there was an identified specific need in patients who were not responding to warfarin innovator product, right? Those medicines were showing benefit in that class of patients. So it's registrable, though not interchangeable with the innovator product. Now, for a generic product to be interchangeable with an innovator product, we consider that they must be therapeutically equivalent to the reference product, that's the innovator product. Pharmaceutically equivalent, right, may not be therapeutically equivalent. We know that because bioequivalence is what equates to therapeutic equivalence, right? And only multi-source, that's only generic pharmaceutical products that have been proven to be bioequivalent or therapeutically equivalent are noted as interchangeable. So once a medicine in Nigeria now has been registered by NAFDAC as a generic of that innovator product, right? It's not as if it will bear a new stamp. All it means is that if a product continues to be registered by NAFDAC that contains omeprazole 20 milligram granules, encapsulated granules, then they are generic or interchangeable for Mepracil 20 milligram omeprazole granules. I hope you understand that. As long as NAFDAC has, approved the medicine as a generic, then they become interchangeable with the innovator product, right? So how do we do this in practice? Practice of substituting a product, whether marketed under a trade name or generic name, with an equivalent product, usually a cheaper one containing the same active ingredients, is a professional professional practice under the jurisdiction of the pharmacist. So you as a pharmacist, once you identify that the generic product has met the evidence required by NAFDAC, then you can go ahead under all legal obligations to provide that generic product in place of the innovator product to the patient, right? But then there are still barriers, because a lot of professionals still believe that there is no proof that the generics and the innovators are bioequivalent. And that's usually because of a poor basis of knowledge about their interchangeability. And also, physicians may not know enough and patients are not educated enough to know that this is so, right? Also, because of the complication that comes in, because of the presence of substandard and falsified medicines in Nigeria, we also find that there's a bit of skepticism about the use of innovator and generic products. And this is usually the problem, because sometimes our pharmacies may decide to procure medicines, especially when the pharmacist is not in charge of the procurement. They may decide to procure medicines that are on discount, right? And then they fall into the hands of people who are importing parallel substandard products into the country, right? So it's always best to stick with those suppliers that are verified by NAFDAC and have something called a provenance. Provenance means that they are buying directly from the from the manufacturer, or they are buying directly from the distributor that delivers for the manufacturer. Okay, so that's an important part. Now, implication in practice is that there's low adherence to prescribing by generic names in public health systems. There's high cost of filling medicines and parallel importations, right? And then the dispensing of pharmaceutical equivalents is currently left to the discretion of pharmacists only, rather than having a lot of people advocating for this in order to reduce cost of medicines, right? So pharmacists, you should not buy at discounts in order to avoid legal consequences from infiltration by impostors and fake importers. Select products on the basis of reputation, right? And the fact that these people deliver directly to the distributor, and then make decisions based on product bioequivalence and quality, right? In this way, you are legally covered and you are doing the best for your patient, okay? So take all this in consideration when you're doing generic substitution, okay? Because this just involves choosing generic over innovator products. It's also a part of what you are currently doing with the e-dispensing. So this part will just come as questions. There are no direct practical because you do not ship the products, right? So this is why I'm just describing this part of the practice. Once we have, availability, once we are able to design something around that, we'll definitely let you know. But for now, basically, it's just to describe it, practice implications. Also, note that there have been challenges or arguments that critical drugs like narrow therapeutic index drugs, like warfarin, for example, that they may not be right. They may not be right for interchangeability, but that's not true because there have been randomized control trials, and a review of randomized control trial recently showed that there is no difference, right? Especially for warfarin, there's no difference between the generic and the and the innovator product. For critically ill patients, people think that very old and neonate patients, presence of multiple diseases, that this requires you to use innovative drugs, that's what works. It is not right, or it is not true. Clinical trials have also shown that there is equivalent biological action between generic and innovator products. Same with critical disease, right? So there is no known contraindication for interchangeability in these conditions right now. So you can proceed with confidence as a pharmacist. Okay. So this is a professional responsibility of pharmacists. You're entrusted by the public to select products from manufacturers that offer the best quality at the best price, and we must exercise this based on evidence, right? So we have this professional obligation to provide medicines that are safe, effective, and also affordable to patients. So that ends the first part, which is the generic interchangeability or substitution. Now, the second part is there are some medicines, right, that are generics, and WHO has also looked at it that, for example, carrying out a bioequivalence study for when you're registering pharmaceutical forms, medicines, you have to do bioequivalence study for every dosage change, every alteration in excipient, every change in the compression factor, all of those things. So what the WHO did was looking at how much generic medicine manufacturers were going to spend, and it was going to be a severe waste, right? It was decided that there's a more scientific way to go about identifying if one medicine is bioequivalent to the other, right? Aside from going through the the in vivo, that's in human trials, right? There is a better way, a more cost-effective way to go around this using just a bench research. And so it was decided that we could classify medicines, right? Class 1, medicines with high solubility and high permeability will be regarded as class 1. In class 2, medicines with low solubility but high permeability as such. And then in class 3, high solubility but low permeability. For classes 1, 2, and 3, in most cases, just bench work is required to prove bioequivalence. But for class 4, medicines with low solubility and low permeability, you may be required to carry out what's it called, a in vivo trial rather than in vitro, because in vitro trials rest on solubility and permeability, right? So where both are low, right, you may require to do human trials rather than in vitro bioequivalence studies. Okay, and this is usually done on finished pharmaceutical product, right? So you provide the finished pharmaceutical product, carry out an independent in vitro bioequivalence test, submit your results to NAFDAC, and then including samples as well to NAFDAC. NAFDAC also tests. So based on the independent test, NAFDAC may give you marketing authorization, but NAFDAC will also do a test on the samples you have submitted to confirm these results that you've submitted, right? And take professional action where regulatory action where required. So if it doesn't meet up, they stop your marketing authorization, and where it meets up, they allow you to continue marketing. So remember, high solubility is defined as an API is considered highly soluble when the highest single therapeutic dose, or as defined by the labeling for the innovator product, is soluble in 250 mL or less of aqueous media over the range of pH 1.2 to 6.8, right? That's acidic. The pH solubility profile of the API should be determined at this temperature, and minimum of 3 tests is recommended. For high permeability, an API is considered highly permeable when so you also you need to usually what's it called? Each manufacturer doesn't need to carry this out. This is done by the API manufacturer, right? And once the API manufacturer provides you, right, they provide you documents proving this under the dossier, the digital dossier that is provided when you are doing your registration. So your API manufacturer must be a WHO pre-qualified manufacturer. There are very few of them, but most of them are already WHO pre-qualified, except in cases where you go to the black market, right? When you do that, you will not be able to provide that dossier to NAFDAC, and NAFDAC will. That dossier, you cannot go and copy it online and copy and paste, or even when it's supplied to you by the black market, it is not acceptable because it must come directly from the API manufacturer, and there are ways that process is confirmed by NAFDAC. For example, they will include all the and the grades of all the equipment that are involved in the manufacturing of that API, the last time they were serviced. And because all of these things are also documented, right, under current good manufacturing practice with the WHO and with regulators, the NAFDAC as an agency is able to look at these data in live view, right, and identify if you're actually providing nonsense to our market or not. So this is possible. This has been possible in the last 5 years, but it was not possible before the last 7 years, but it was not possible before that. High permeability is also determined as such. Now, how do we determine the solution characteristics for a BCS biowaiver application, right? Now, for that, BCS is that classification that we talked about earlier on, this classification, right? We use it for something called a biowaiver. It means that when you're, like I explained earlier on, once your API passes this first tree, then you can get a biowaiver to only do in vitro testing. Now, how do you determine the solution characteristic? For exemption from this, an immediate release generic product should exhibit very rapid or rapid. So what is very rapid or rapid? That's between BCS class 1 and 3. For very rapid, right, generic product is considered to be this when not less than 85% of the labeled amount of the API dissolves in 15 minutes at this temperature, that's room temperature, 37 plus or minus 1 degree C, using a paddle apparatus at 75 rotations per minute, or the basket apparatus. we used to have both of these, both the basket and the paddle apparatus. But right now, the paddle apparatus that we have is not working, but you can view it in the clinical pharmacy postgraduate lab. And I think there's one, the paddle. I think there's also another paddle apparatus available at the what's the name of that other lab in the faculty? I think it's called I think it's called general, no, not general lab. There's a lab that doesn't belong to any of the departments in the faculty. All the departments have access to that laboratory. It's like, oh, good, central lab. Okay, there's one in the central lab, and it's also a paddle apparatus, right? So you test it at 75 rotations per minute in a volume of 900 mils or less in each of the following media. You do at highly acidic, moderately acidic, and then at phosphate buffer, right? Once it is tested at all of these levels, you must have minimum of 85% dissolution in 15 minutes. This is just about the buffers, the media. Now, for rapidly dissolving, no less than 85% dissolves in 30 minutes at same temperature, same RPM, at 1.2 HCl, 4.5 acetate, 6.8 buffer. Surfactants should not be used in dissolution media. Enzymes may be used if pharmaceutical product contains gelatin. All these are just specifications, and usually it depends on the type of what's it called? Depends on the type of the type of excipients present in your formulation. Now, how do medicines qualify for BCS biowaiver? Once you prove that it's soluble, right? The solubility and intestinal permeability of the API must be submitted to the regulator, and then, of course, similarity of the solution profiles between generic and comparator, that's described in the last page, right? In 1.2, 4.5, and 6.8 media should be submitted. Excipient, list of all the excipients used must be submitted. And then, of course, the risk for an incorrect biowaiver decision in terms of therapeutic index and clinical indications for the API are also considered, right? If there are clinical proofs that there may be there's narrow therapeutic index, right, for the API, then it is carefully considered, and clinical evidence is also included in taking a judgment about that drug. Now, the excipients are also evaluated. In all cases, well-established excipients must be available in usual amounts, right? If you're adding one excipient in larger quantity than is usually used, for example, if your disintegrant or your binder is more than 5 to 10% of your entire total tablet weight, then of course that will raise eyebrows at what is happening. It's either your formulation is bad, or there's something wrong with the way you're compressing your tablet, right? Excipients that may affect bioavailability must be identified, right? And there should be, if that is different, in your tablets to that of the innovator product, you must be able to provide reasons why that is so. For biowaivers for products containing class 1, right, there's flexibility because those are very highly soluble, except it contains one of these mentioned in number 2 above, right? Usually you will be you can have as many your excipients can be different from the innovator as much as you like. For those ones containing class 3, right, which is at the tail end, all the excipients should be qualitatively the same and quantitatively similar to that of the innovator. because if there are, large differences, it can further worsen what we have in terms of the high solubility may be affected, which will be compounded by the low permeability, resulting into very different bioavailability of the medicines. Now, in vitro testing can also be based on proportional dose of the formulation, right? And this is basically just one of the methods for testing, but this is not as common as the other method, which is recommended for conducting and assessing the comparative dissolution profile. So this is the main method, right? The dissolution measurements of two finished pharmaceutical products, FPPs, right, should be made under the same test conditions. Minimum of sampling 3 times, right, should be included in the test. So over that 30 minutes, you must sample. So immediately you put the tablets in, you are drying your sample from the PADU solution. Then 5 minutes later, you draw again, 10 minutes to draw, 15 minutes to draw, 20 minutes to draw. So minimum of 3 draws are taken, right, up until and then compared, okay? The 15-minute time point is critical to determine whether a product is very rapidly dissolving and to determine whether the similarity factor needs to be calculated. The similarity factor is only calculated when there appears to be a difference at the 15-minute time interval to amount of drug in solution in the PADU or the basket test at 15 minutes, right, under the RPM and media, right? So for extended release products, right, this is different. Of course, extended release products are different from immediate release products. So for extended release products, you should cover the entire period that is expected to for the formulation to release its content. So in addition to the earlier time points that we have 5, 10, 15, 20, 30, 45, and 1 hour, right? You should sample 2 hours, 3 hours, 5 hours, and 8 hours, right, for a 12-hour release. So up until 12 hours, you must continue to sample and then do test intervals for longer duration. If, for example, the release profile shows that it's the controlled release medicine releases over 24 hours. There are some medicines like that. For example, Epilim Chrono, right? Epilim Chrono would release over 24 hours, right? For such medicines, including nifedipine retard or Nifecad XL, right? They are 24-hour release profiles. So for such medicines, of course, you must sample until the end dates, end release time as planned by the manufacturer, right? So those studies should be performed in at least 3 media, like we mentioned earlier on, 1.2, 4.5, and 6.8 buffer, right? And the recommended buffers are stated in the pharmacopoeia, right? If both the test and reference products show more than 85% dissolution at critical point, 15 minutes, then the profiles are considered similar. Otherwise, if they are not similar at 15 minutes, then you perform this calculation, which is the similarity factor, right? Where RT and TT are the mean percent API dissolved in reference, and that's reference RT, and the TT is the test product or the generic product, right, at that time point. So you calculate, and then if the F2 value is between 50 and 100, that's your F2 you get between 50 and 100, then it suggests that the two dissolution profiles are similar. But if it is less than that, then they are dissimilar, right? A maximum of 1 time point should be considered after 85% dissolution of the reference product has been reached, so that you do not over sample, and then you spend so much money, running your test. So these are the remaining definitions for what to consider for your similarity factor, and how to report it to the innovator. So with that, we come to the end of this description of how to perform the comparative dissolution profile. So it's very straightforward, and I'm hoping that over the next, maybe before you guys graduate, we're able to fix the machines and then do at least one demonstration, to help you see. The solution machine itself, it's not like it's very expensive. I think the challenge has been with funding that has really gone down in terms of funding from the federal government for practical endeavors. So we hope that very soon we can have this done much more practically. But so far, this is what you need to understand. This is how to test for bioequivalence of products in class 1 to class 3, and how to prove their bioequivalence to the regulator NAFDAC. So far in this class, we have covered 2 main topics. One, interchangeability of generic products. We've proven that once generics are registered in Nigeria, then they are interchangeable with the innovator product. We've talked about the challenges in practice. We've said the main challenge is the ignorance of physicians and patients. And we've also mentioned that another challenge is the incursion of substandard and falsified medicines into our legitimate distribution, drug distribution lines that have caused a loss of confidence. But then that incursion is still less than 15%, right? Which means that 1 out of 10 are, substandard. And the way to identify a very simple way, or a very simple litmus test, is if most of the discounts that happen, right, are outside the authorized pharmaceutical distribution chain. Don't procure medicines. If your pharmacy does not procure medicines outside of the established distribution line, you will not be buying fake and substandard products. Okay, so it's important that we stick, as pharmacists, we stick to the registered distribution line and only stock from professional sources. And that will help us avoid SF medicines. On the second hand, we also talked about how we talked about the why of establishing bioequivalence for class 1 to 3 medicines on the BCS scale, and how to carry out the dissolution test that leads to the regulator accepting that the generic medicine is bioequivalent to the innovator brand. And we described the dissolution process, and how if at 15 minutes, which is a critical time, if at 15 minutes the dissolution is different for both products, you can calculate the F2, the similarity factor. And if the similarity factor is less than 50%, then you cannot say that the dissolution profiles are similar. But if it is between 50 and 100, then you can say for sure that the dissolution profiles are similar. I hope with these I've been able to describe to you the evidence basis and the professional reasons for generic substitution, and secondly, also described efficiently to you the solution tests that help us prove bioequivalence, or the in vitro tests that help us prove bioequivalence for class 1 to 3 BCS class medications. If there are any questions, please let's have them quickly so that I can let you go for the day. I will also share the recording with you after this class. Any questions? Okay, no questions. I want to say thank you to everyone. Thanks for attending this class. Like I said, I'll share the recording and the slide notes shortly after this class. Thank you very much and have a pleasant day. Okay, sorry. Okay. Not questions. All right. Thank you too. Thanks, everyone. Bye.